First, we'll discuss normal synthesis, function, and regulation of adrenal gland hormones. The adrenal glands are located on top of the kidneys but are completely distinct organs from the kidneys. Each adrenal gland is composed of an outer layer or a cortex and an inner layer or a medulla. And the adrenal cortex is responsible primarily for the secretion of aldosterone, cortisol, and several androgens. And all of these adrenal cortex hormones are synthesized from cholesterol. The adrenal medulla is composed of entirely different tissue, namely nervous system tissue, and it's responsible primarily for the secretion of catecholamines, such as epinephrine and norepinephrine, and these hormones are synthesized from tyrosine. This shows a table of the same information we just discussed and also expands on that information. So note that the adrenal cortex is divided into these three zones, and the zona glomerulosa is responsible for aldosterone production. Aldosterone is the main type of mineralocorticoid produced, although there are some other intermediaries produced in the glomerulosa. The zona fasciculata is responsible for cortisol production and other glucocorticoid production. And the zona reticularis, which is the deepest part of the cortex, is responsible for producing androgens, including dihydroepiandrosterone or DHEA and androstenedione. The primary targets of each hormone are listed, their hormonal effects, and their stimulatory factors. So this provides a nice summary for review. So now we'll discuss each of these topics in detail for each hormone individually, beginning with aldosterone. So aldosterone is the first adrenal cortex hormone that we'll discuss, and it's produced in the zona glomerulosa, which is the outermost portion of the adrenal cortex. And that's where the necessary enzymes are to convert cholesterol all the way to aldosterone. And these processes and the enzymes involved are pictured here in this dotted line in the middle and right-hand portions that aren't enclosed in that box will be discussed in subsequent portions. Once aldosterone is produced, its main function occurs within the renal collecting duct, primarily in the principal cells, which are aldosterone's main target cells. So aldosterone, being a steroid hormone, enters the target cell, binds with its receptor, and once the hormone binds its receptor, it induces increases in specific gene transcription, and the end result is an upregulation of channels and processes that allow for reabsorption of sodium from the tubular fluid. So the ultimate end effects of aldosterone are to increase sodium reabsorption from the tubular fluid and also to promote potassium secretion into the tubular fluid, followed by excretion in the urine from the body. And aldosterone is regulated primarily by the kidney. So the kidney produces renin, which promotes the conversion of angiotensinogen and alpha-2 globulin to angiotensin 1. Angiotensin 1 is converted to angiotensin 2 by angiotensin converting enzyme or ACE, and angiotensin 2 binds to its receptors on the glomerulosa cells within the adrenal cortex. And so that triggers a host of enzymatic reactions that we just looked at, which ultimately results in aldosterone production and secretion. So aldosterone, as we already saw, leads to renal sodium reabsorption or retention, and water follows so that both sodium and water are retained in the presence of aldosterone production. This process ultimately increases extracellular fluid volume, and it follows then that aldosterone would be inhibited and activated by factors related to fluid and salt. So inhibiting factors include increased extracellular fluid volume and increased arterial pressure. So both of these factors will decrease renin production from the kidney and thereby decrease aldosterone. However, when the body's in need of extra sodium and fluid, meaning conditions such as decreased extracellular fluid volume, decreased arterial pressure or blood pressure, and states of general sodium depletion, renin will be upregulated and ultimately cause increased aldosterone. So let's turn now to cortisol, which is synthesized in the fasciculata or middle layer of the adrenal cortex. And this is the zone that contains the necessary enzymes for converting cholesterol all the way to cortisol. And for example, this 17 hydroxylase, which is present in the fasciculata, can convert some of the normal aldosterone precursors to cortisol precursors. Note also that the fasciculata does not contain the 18 hydroxylase, and therefore it's not capable of synthesizing aldosterone. Cortisol serves several critical and life-sustaining functions, so it's a steroid hormone just like aldosterone is, and it binds to specific glucocorticoid receptors inside most cell types. The main effects are listed here with an acronym that will hopefully help remember some of the effects. The two B's stand for blood pressure and bone formation. The two I's stand for inflammation and immune function. And the G stands for gluconeogenesis, or production of blood sugar, as well as lipolysis and proteolysis, which refers to breakdown of fat and protein to convert to immediately usable energy. And you can see that there are these two up arrows, meaning that blood pressure and glucose production are increased by cortisol, while the more housekeeping type of functions like bone formation, inflammation, and immune function are suppressed by cortisol. The regulation of cortisol is very different from the regulation of aldosterone. So cortisol production is controlled by the pituitary, as you might remember, when CRH from the hypothalamus stimulates ACTH from the pituitary. And this ACTH will then bind to its receptors on the fasciculata cells within the adrenal cortex. And ACTH has several specific actions, including stimulation of uptake of lipoprotein in fasciculata cells to serve as a substrate for making cortisol. And it also stimulates this rate-limiting step in the production of cortisol, the conversion of cholesterol to pregnenolone. Once cortisol is produced, it exerts negative feedback effects on both CRH and ACTH. The third hormone from the adrenal cortex is actually a set of hormones, the androgens, which are produced in the zona reticularis, or the deepest layer of the adrenal cortex. And again, the enzymes necessary for making these androgens, known as DHEA and androstenedione, are specifically present in the zona reticularis. So testosterone and estrogens are listed below the adrenal androgens as a reminder that there are other types of androgens in the body. Testosterone is primarily produced by testes, and the estrogens are mainly produced by ovaries. And testosterone can also be converted to estrogens by the aromatase enzyme in several tissues throughout the body. The function of androgens differs between females and males. In females, the adrenal androgens promote development of pubic and axillary hair, and the hormones also contribute to libido or sex drive. In males, the adrenal androgens have a less significant role generally, especially after puberty. And this is because males make testosterone from the testicles, which is the major androgen present. There are pathologic conditions where excess adrenal androgens can be produced. These can be significant clinically in females and prepubertal males because signs of virilization and masculinization can be seen inappropriately. Some examples that will be discussed in the clinical sessions include congenital adrenal hyperplasia, or CAH, and androgen-producing adrenal tumors. And androgens from the adrenal gland are also regulated to some extent by ACTH from the pituitary. And there are ACTH receptors located on the membrane of the reticularis cells. And ACTH binding its receptors can promote the conversion of cholesterol to androgens like DHEA. However, the main negative feedback effect on ACTH still occurs from cortisol. Now that we've discussed the adrenal cortex hormones, aldosterone, cortisol, and androgens, we'll turn to the adrenal medulla. And this is where the catecholamines are produced. The three hormones located below the dashed line are catecholamines, namely dopamine, norepinephrine, and epinephrine. And these are all synthesized from tyrosine as a precursor through a series of enzymatic steps which can occur in the adrenal medulla and also in other sympathetic nervous system tissue. So it should be noted that the last enzyme in the cascade, the one that converts norepinephrine to epinephrine, is only located in the adrenal medulla, which means that epinephrine will not be made in other sites of the sympathetic nervous system. These compounds in the red rectangles are the breakdown products of each of these catecholamines. Later, we'll review how either the catecholamines themselves or their breakdown products can be measured. The function of the catecholamines and their main effects are seen primarily on the cardiovascular system and the area of general metabolism. So cardiovascular effects vary by the specific catecholamine depending on where the receptors are located. So this first table summarizes the target receptor for dopamine, norepinephrine, and epinephrine with the actions and physiologic responses listed. Dopamine causes vasodilation in the heart, kidney, and mesentery, which results in an increased blood flow to vascular beds. Norepinephrine or adrenaline binds alpha receptors and beta-1 receptors, resulting in intense vasoconstriction, which causes an increase in peripheral vascular resistance. Epinephrine binds to alpha receptors as well as beta-2 receptors, and this causes vasodilation in the skeletal muscle, but vasoconstriction in the skin or the viscera. And epinephrine also stimulates cardiac muscle cells. And the end effect is to be neutral on peripheral vascular resistance, but to increase cardiac output and heart rate. The metabolic effects of the catecholamines are to increase energy stores for immediate use via glycogenolysis and lipolysis. And the catecholamines also suppress insulin release because insulin is, in a sense, a storage hormone that would inhibit the release of immediate uses of energy. So catecholamines can then be summarized as fight or flight hormones because they mimic the effect of the sympathetic nervous system discharge. Catecholamines are regulated from the brain and nervous system as you might expect. Stressors and other signals will increase nerve stimulation to the adrenal medulla, causing production of the catecholamines via tyrosine conversion to the products we mentioned previously. Cortisol production is also shown here to let us compare and contrast the two hormones. And while both catecholamines and cortisol are considered stress hormones or flight or fight hormones, the catecholamines are produced much more rapidly in response to a stress or some type of stimulus. And that's because the catecholamines are preformed and can simply be secreted much like a peptide hormone, while cortisol is a steroid hormone that first has to be synthesized once the fasciculata cell is stimulated by ACTH from the pituitary gland.